Hey there. So today we are going to take a look at our incomplete and codominant notes. So I'm going to start with packet page 87, which is our incomplete dominant notes. So we already know about regular dominance, basically meaning if you have an uppercase letter, you know that that trait is going to be expressed, and you need two lowercase or recessive letters or alleles in order for that version to be expressed. So that's not always true, right? Not every trait follows that simple rule. So some alleles are neither dominant or recessive, basically meaning some of these traits are controlled by multiple alleles or multiple genes. And so today we're going to learn about two of those types of other dominance rules. The first of which is incomplete dominance. So incomplete dominance. So this is when one allele is not completely dominant over the other allele. So think blending. What we see in incomplete dominance is the heterozygous phenotype is somewhere in between the two homozygous phenotypes. So up until now, we've said big T is tall, little t is short. If you are heterozygous dominant, big T, little t, you would be tall. But that's not necessarily true. So we're going to look at an example of what we mean by incomplete dominance. So I'm just going to kind of roughly half. There we go. Just kind of split this in half because we're going to do two examples. So imagine you have a flower called a snapdragon flower. In snapdragons, the flower color is incompletely dominant. So basically, right, if we think about our alleles, red flower color is dominant. So I would have, two big R's. White petal color is recessive. So I would have two W's. And then if I crossed a red snapdragon and a white snapdragon, I would end up with a blending. And if you think about an art class, right, what color do you get when you blend red and white? Well, you get pink. So the heterozygous genotype here would be RW, right, to give us pink. So if we were to cross, a red flower with a white flower, all the offspring will be pink, right? Because red crossed with white gives me that heterozygous form for pink. So what percent of the offspring will be white if a pink flower is crossed with a white flower? All right, so that's our question. So we need to write out our parent genotypes. So we've got what percentage would be white? We have pink crossed with white. So the genotype for pink is RW being crossed with white, big W, big W. So now I can set up my Punnett square. I need to place one of my parent genotypes at the top and the other one on the left side. I'm going to place the pink genotype parent at the top of the Punnett square because I feel like it, and then the white parent over here on the left side. I'm now going to distribute my alleles, and I need to remember that the dominant allele always goes first, and red is technically right dominant to white, and then of course together you blend them. So when I distribute my alleles, I would put the R and then the W, two W's, RW, two W's. So the question is, what percent of the offspring will be white? Well, 2 out of 4 of my Punnett square boxes show me white. So my correct answer is 50% will be white. So that is how incomplete dominance works. But let's do another example just to make sure. So let's use sunflowers, right? Kind of sticking with that flower theme. But the alleles, right, for sunflower, if you are tall, right, 2 big T's. If you're short, right, 2 big S's. But if they blend, if I cross a tall and a short sunflower, I get a medium sunflower. So that would be, oops, sorry, I wrote that backwards. I apologize. TS. Right, so that is my blending, or my incomplete dominant medium version for sunflowers. So my question to you, if 2 medium sunflowers are crossed, what percent of the offspring will be medium height? Question mark. So the first thing you do is write out my parent alleles, and they tell me that they're both medium. So both heterozygous dominant. I can now draw my Punnett square, and then need to place my parent alleles. Now I can distribute those alleles to see what my offspring will be. S, T, S, S, S. So they want to know what percent of the offspring will be medium? Well, in order to be medium, you have to be heterozygous, and I only see 2 out of 4 of these boxes. So again, 50% would be medium height. So that's our notes for our actual packet pages. You would need to go into the Nearpod and complete the notes from the Nearpod underneath that. So just make sure you check the Canvas agenda to see what you need to do there. So let's take a look now at the other side, right? And let's talk about codominance. So here for our codominance notes, again, sometimes alleles, they're not always just dominant or just recessive, but also there's no blending. Remember, if it's blending, that means we have incomplete dominance, right? Red mixed with white to give me pink, tall mixed with short to make medium. But instead, if an organism has 2 different alleles, we can have both of these traits show up, and this is called codominance. So both of the versions are being expressed in that organism. So let's take a look at our first example. In certain types of chickens, the allele for black feathers, B, is codominant with the allele for white feathers, W. And so heterozygous chickens have speckled black and white feathers. So they show us our genotype and our phenotype. So, our heterozygous would have both black and white speckled feathers, right? They have spots. So we've kind of got 2 examples here. we want to know what is the probability of a baby chick having black feathers, white feathers, or black and white feathers based on these two parent genotypes. So one parent has black feathers and the other parent has white feathers. So our parent alleles and our Punnett square have already been set up for us. All I need to do is distribute the alleles. So I'm going to go ahead and do that. There we go. So what is the probability of a baby chick having black feathers? Well, I only need one version of the allele to have black feathers, and I see that in all four boxes. So I would say there's a 100% chance of that happening. And then what's the probability of white feathers? Well, in order to have white feathers, I would need two copies of that allele. That did not happen at all, so 0%. And then black and white feathers. Oh, that's 100%. I apologize. This should be 0%. See, look at that. I made a mistake. I'll just go ahead and scratch all that out. 0%. But I caught my mistake, and that's what matters. The only way to have black feathers is to have two capital Bs. None of these offspring would have black. They would all have black and white feathers, meaning they are codominant. So I do apologize for that mistake. Please let me know if you have any questions, because I did accidentally say that incorrectly, but I fixed it, right? None of them have black feathers. None of the chicks would have white feathers. They would all be speckled. So the second example, this parent has speckled feathers and this parent has speckled feathers. So I'm going to distribute my alleles again. Hopefully I won't make any mistakes this time. So what is the probability of a baby chick having black feathers? Okay, I do see two uppercase B's together. Now there's only one square, so that would be 25%. The probability of having white feathers right here, so another square is 25%. And the probability of a baby chick having black and white feathers, that's the other two squares. So we're looking at 50%. So one easy way to check yourself to make sure you have your probabilities correctly is that it should always total 100. And that's how I caught my mistake over here, is because I saw 100, 0, 100. And I was like, 200%, that doesn't work. But I did manage to catch that. So that's what we mean by codominance. And that's an easy example. Let's take a look at example 2 over here on the right-hand side, talking about blood type. So many cells in the human body have protein tags called antigens on them, so that our body can identify which cells in our body are self cells and which cells are foreign invaders. Our blood cells can have these antigens on them as well. There are two main types of antigens or tags that can be on our blood cells: A antigen and B antigen. Someone who has no antigens on their blood cells is said to have type O blood. All right, so I have A antigens, B antigens, or I can have neither of those. So I kind of have three options there. So the protein tag that antibodies can bind to and trigger an immune response, right? That is an antigen, right? So we're kind of talking a little bit about the immune system here and what the body would recognize as itself or as a foreign invader. Proteins that recognize and bind to antigens and can destroy these foreign invaders, right? Those are antibodies. So talking about blood type, so draw a red blood cell that would be found in a person with type A blood. Okay, so if we draw a red blood cell for someone with type A, so let's say, right, here's our red blood cell, and maybe it has these kind of A-looking spikes kind of coming off of it. So those are the antigens, right, for A. And then a red blood cell would be for someone with type O, right? Their red blood cell would be empty. because they don't have any antigens at all. And then, of course, someone with type B, their antigens would just look different than the A ones. Maybe they're little squiggly lines or something. But basically, A or B blood types have the antigens sticking off of them. Anyone with blood type O, their blood cell is basically empty. So if we look at our practice problem here, so what are the possible blood types for their children? So with blood types, we are talking about codominance, because I can be blood type A, I can be blood type B, and I can also be blood type AB. Let me zoom in here a little bit on this side. So when we're looking at the alleles for blood type, if I have an allele from blood type A and an allele from blood type B, I can be blood type AB. If I'm completely missing those alleles, then I would be blood type O, which is completely recessive. So really, blood type is very unique because it has 3 alleles. I'll write them up here. So you can be blood type A, blood type B, right, or recessive. So I've got an uppercase I, a lowercase I. So we've got actually 3 different alleles that we're working with for blood type, whereas for everything else, we've only had 2, right? Big A, big B, little b. But blood type, I've got 3. So let's go ahead, let's distribute our alleles here to for our first practice problem. So my uppercase I, A, uppercase I, B, A, B, A, B, A, B. So the question was, what are the possible blood types for their children? Well, in all 4 squares, I see AB. So the only possible blood type is blood type AB. Ooh, that kind of weird. That's the only possible blood type. And then our second example over here. So again, parent genotypes are already here. This parent has blood type A. They're just heterozygous for blood type A. And then this parent is blood type B. So let's see what their possible blood types are for their children. So A, B. Oh, I wrote that a little backwards, but that's okay. So the possible blood types for their children, wow, is all of them. They can have a child that's blood type A, heterozygous, blood type B, heterozygous, running out of space, blood type AB, right, and also blood type O, right, because 2 recessive alleles gives me blood type O. So this couple, right, and we're not saying that if they have 4 children, all 4 children will have a different blood type. We're just saying there is a 25% chance their children will be blood type O, 25% chance blood type A, 25% chance blood type B, or 25% blood type AB. Last thing about blood type. Why is type O considered the universal donor? Well, it's because blood type O has no antigens. And so remember, we said antigens act like little tags, and your body can identify those tags as something that is you or as something that is a foreign invader, right? And just like bacteria, if it's a foreign invader, your body's going to attack it. You'll have an immune response, which we would call getting sick. So if you ever you need a blood transfusion, if we don't know your blood type, you will get blood type O. Because it is missing those antigens, your body will not attack that blood, and you'll be able to accept it, and you'll survive, any blood loss. We'll be able to replace it. And then why is type AB considered the universal recipient? Basically because immune systems with AB blood are used to seeing A's and B's. So they are used to seeing both types of antigens. Let me turn the paper. Here we go. Antigens. There we go. So, for example, I believe I'm O positive. So very close to being O negative and the universal donor. Um, but because I'm O positive, right, my blood type is O, I don't have any of those antigens on them. So if I were to receive A or B blood, my body would not it would not recognize those A and B antigens. It would see them as foreign invaders, and I would reject that blood, and that could be life threatening. So, right, that's what we kind of mean when we talk about a universal donor or a universal recipient. And just like I said a second ago, make sure that you check out the Nearpod. It's the same Nearpod as the Incomplete Dominant notes. You'll have to handwrite in your notes from that Nearpod into your notebook. But as far as our packet pages are concerned, this is it for our notes. If you're curious about anything with blood type, or if you have any questions about incomplete or codominant inheritance, please just make sure to let me know. Okay? See you next time.